Hello, family. I am Judith Constantine with my pharmacognosy-related subjects. Please welcome to my channel. And here is plant tissues of diagnostic importance, part 5. We have done part 1, part 2, 3, and 4, and this is part 5. In this part 5, we are going to discuss about vascular tissues. Remember, we said that the plant tissues of diagnostic importance include those of the vascular tissues, includes the epidermal tissue system and includes the ground tissue. So we have done the epidermal tissue system where we have seen some things about the epidermis. We have seen about the the trichomes associated with epidermal cells. We have seen about stomata associated with epidermal cells. We have seen how epidermal cells can be specialized to perform specific functions, like what we have seen with subsidiary cells. We have seen with the guard cells. And we have seen the types of stomata, we have seen the types of trichomes, and how can these be used to identify various sources of medicinal plants. And please welcome to part 5 of the plant tissues of diagnostic importance. The vascular tissues in plants are the essential transport system, primarily composed of the xylem, which is responsible for moving water and minerals up, and the phloem, which is responsible for moving sugars and nutrients down and around the plant. They work together in structures called vascular bundles. I hope you all know about these vascular bundles, which are there for support and distribution, allowing advanced plants to grow larger and thrive. The xylem uses the hollow cells which are dead. They are called trichots or vessel elements. And for efficient water flow, we need those trichots and vessel elements. Also, the phloem uses sieve tubes to transport food. We are not going to dig much into these trichots, vessel elements, and sieve tubes. What we want is to see how we can use the xylem and phloem to differentiate plants at bigger taxonomic levels like monocotyledons, dicotyledons, angiosperms, and gymnosperms. So vascular tissues are important diagnostic tools in plants because their structure, their arrangement, and the condition provide crucial information about the plant species identification, growth, overall health, and response to environmental stressors and pathogens. So plant species can often be identified or classified at higher taxonomic levels, like I said earlier. This has been already said in the previous slides. So monocotyledons. In monocotyledons, the vascular bundles are typically scattered throughout the stem. And in the roots, they form a ring around the central pith. We are going to see this in the images. And in dicots, the vascular bundles in the stem are usually arranged in a distinct ring. They do not have a central pith like that on the monocot roots, so they can be easily differentiated from the monocot roots we are going to see as we move. And the xylem often follows a central X or a star shape, just like my fingers here. The xylem appears in a star shape or an X, and the phloems will be found in between these fingers, in between these arms of the star. We are going to see as we move, and we will get to understand each other. So the leaves typically have branched ornate veins in dicots, and the leaves have parallel veins in the monocots. This is what I was talking about in the previous slide. You see, look at the dicot stem and the monocot stem. We see that in the monocot stem, the vascular bundles are scattered. These are vascular bundles scattered throughout the ground tissue in monocot stem. In dicot stem, the vascular bundles are arranged around a distinct ring, but remember they do not have a central pith like what we will see with the monocot ones. These are stems. This is the cross-section view of dicot and monocot stem. This is what we expect to see under the microscope. This is the image from the microscopic images. You see, this is a dicot stem where the vascular bundles are arranged in a distinct ring. And these are vascular bundles. In monocot stem, they are just scattered throughout the ground tissue. So that's it. And in dicot roots now, the dicot roots, we said that the vascular bundles, especially the xylem, forms an X at the center or a star shape. We are going to see how the star shape appears, and this is an X shape. And the phloem is between the arms of the X or the star. It's like between the fingers. That's why I showed you by using my fingers. Like, assume this is a xylem, and the phloem will be found in between the fingers. And this is the monocot root. Remember, we said that in dicot stem, the vascular bundles are arranged around in a distinct ring. In monocot root, the vascular bundles are around in a cycle around a central pith. This is the central pith, and these are xylems. These tiny dots are xylems, and in between xylems we find phloem. In between xylems we find the phloem. So that is how the monocot roots and dicot roots appear under the microscope. Here is the microscope image. You see, this is an X shape. We said the X shape is for the xylem. And in between here, we have the phloem. And the monocot root, we have the xylem and the phloem between the xylem around the central pith. So this is the central pith. Okay. That's it. And here, this is the dicot root. Remember, we said that in the dicot root, the xylem can either form a star shape or can form an X shape. And this is the star shape that has been formed. This star shape is the xylem with its fingers here, and between the fingers we find the phloem. That's it. So, what are these? Remember, those are monocot stem and dicot stem. Why? Because in monocot stem, the vascular bundles are scattered throughout the ground tissue, and in dicot stem, we expect the vascular bundles to be arranged around the distinct ring. And that one, that is obvious, a star shape. We said the star shape is for a dicot root, where this star is a xylem, and in between the xylem we find the phloem. So in woody plants now, the xylem is crucial for determining age and past environmental conditions through the analysis of growth rings, also known as annual rings. This process is normally done by, this process is done by dendrochronologists. The dendrochronologists are the people who deal with the science or technique of dating events, dating environmental stresses, and they deal with archaeological facts about plants. They can use the patterns of annual rings in timber and tree trunks to determine the age of that particular plant. And the wood is the secondary xylem. Remember, we said the xylem in bigger trees can turn into a wood. So scientists who study tree rings, also known as dendrochronologists, analyze the patterns in xylem growth rings to determine age by counting the total number of rings So by counting the total number of rings, the age of the tree, the age of the tree at the height where the sample was taken can be determined by counting the total number of annual rings. And to determine past climatic conditions, we deal with the width of these vascular bundles, of the width of the xylem, and we deal with the characteristics of the xylem. So each ring will be examined for any changes that will help us determine certain environmental conditions in that specific year. And for example, wider rings tell us that we had favorable growth conditions or we have a sufficient rainfall along a growing season. And narrow rings often signify stressful years such as droughts, extreme temperatures, insect infestations, or nutrient deficiencies, and the like. And about historical events, if we find scars or sudden changes in growth patterns, this indicates forest fires, floods, or insect outbreaks, and the like. So, by cross-referencing ring patterns from multiple trees in a region, scientists can create long-term climate chronologies extending data from 1,000 years. We're not going to dig into dendrochronology, that is not necessary here, but this was just for you guys to appreciate the role of these vascular bundles when it comes to plant identification. For example, we count the rings from here, first growth year. We keep counting until we get the number of years the tree has lived. But by looking at the width here, look here, this is bigger width. By looking at the width, this tells us that these were favorable conditions. We had rain seasons here. By looking at the width here, this is smaller. This tells us that there's a stressful condition. Maybe it was the dry seasons. By looking at the scars, this is a scar. It can tell us that the scar maybe was from forest fire and the like. So that's how it works. Now, the next part will be on ground tissues. That will be part 6. So stay tuned to wait for ground tissues, and I really thank you guys for keep watching these videos. Please, if you have any comment, if you have any reference material that you think will be important for us, if you have a question, if you feel that we can discuss or pose out something, please feel free to do that. We can keep ourselves updated as the knowledge increases in this area. Thank you, and please subscribe, share, comment, and see you next time. Bye.